Picture this. You sit down for your exam and you confidently write down DNA polymerase builds DNA. Sounds perfectly accurate, right? Well, relying on generic facts like that is the exact mistake that is costing top students their grades. Welcome to this explainer. Today, we're mastering section 6.1, semi-conservative DNA replication, and we're going to look specifically at the roles of DNA polymerase and ligase for the Cambridge AS and A-Level Biology 9700 syllabus. I'm going to show you exactly what Cambridge examiners are actually looking for and how you can stop losing the marks you totally deserve. Listen to this classic example of that devastating mistake. Quote, DNA polymerase adds nucleotides to the new strand and ligase joins the fragments together. End quote. You know, most students write this. They feel pretty confident and they just move on. They genuinely think they've nailed the biology. But I'm telling you, this generic textbook-style answer is a massive trap. It is literally the classic mistake that costs candidates marks before they even realize what hit them. So what does that answer actually get you on the exam? A big, fat 0. Nothing. If your answer isn't tied directly to the specific scenario they gave you in the question, you lose the marks. You simply can't just memorize textbook sentences anymore. You have to actually apply those concepts to totally unfamiliar situations. Okay, let's dive into this and secure a quick win by anchoring our core biological principle, semi-conservative DNA replication. What is it? Basically, it's the process where each new DNA molecule ends up consisting of one original parent strand and one brand newly synthesized strand. This right here is the absolute foundation. Top-performing students use this foundational knowledge to build logical, highly structured reasoning in their exam answers. Now, think about our two star players here, because they have very different jobs. DNA polymerase, it acts kind of like a robotic arm, physically building the new strands by adding nucleotides. Meanwhile, DNA ligase acts as your ultimate sealing tool, coming in to close up the gaps. Understanding these distinct roles is absolutely crucial. But, and this is a big but, knowing these facts is just the starting line. It's just the baseline required by the syllabus. To actually get the marks, you need to know how to use them. So what is the hidden examiner clue? Well, in a moment, I'll tell you the exact clue that changes your answer completely. But first, you really need the framework that makes that clue completely obvious to spot in the first place. Sticking around for this means grabbing the very strategy that unlocks the highest mark bands in your Biology 9700 exams. All right, Section 1, the high mark framework, specifically the method mark strategy. Let's jump right into how this works. This is essentially the procedural blueprint that elite candidates use to secure process marks in really complex problems. Remember, in Cambridge exams, marks are heavily awarded for the process of getting to the answer, not just stating the final fact itself. So, how do we break this down? There are 5 mandatory steps. Step 1, deconstruct the question. Ask yourself, what are they actually asking here? Step 2, extract the relevant data from the stimulus materials they gave you. Step 3, select your analytical framework. Step 4, execute that with clear notation. And finally, Step 5, present your final answer using the correct terminology. This exact methodology completely transforms a messy, panicked response into a highly structured, top-tier answer that examiners absolutely love to see. All right, let's play a quick game and put this into practice. Spot the error. I'm going to present 2 different answers. I want you to pause, predict what's wrong with the first one, and spot the error by applying the framework we just talked about. Think about context, and think about precise terminology. Ready? Let's do it. Let's look at the flawed approach versus the perfect approach. On one hand, a student might simply say, ligase joins DNA. It's just way too simple. Now look at the detailed mechanism for a top-tier answer. Ligase forms phosphodiester bonds between Okazaki fragments in the lagging strand scenario provided. Boom! That second answer gets full marks. And why? Because it uses precise scientific terminology tied directly to the scenario provided. Always link every single point to the given scenario. Moving on to our next section, escalating the stakes with statistical analysis. Let's ramp things up a bit. Your exams aren't just about pure theory, right? They demand serious data interpretation and practical skills, often in totally unfamiliar contexts. When you're extracting data, you absolutely must select the correct statistical test. So if you're comparing means, you use the t-test. Got categorical data, you're going to want to use the chi-squared test. And for finding relationships between variables, you use correlation. Top-scoring candidates evaluate the quality of evidence by identifying limitations and assumptions using exactly these tools. It's a total game changer. Section 3, Principal Examiners' Reports and their top 3 warnings. And this brilliantly illustrates what examiners are actually thinking when they grade your paper. We actually researched the Cambridge Principal Examiner reports for this specific topic, and we found the hard data on exactly where students are messing up. Warning number 1. Students constantly forget to include units in their 6.1 semi-conservative DNA replication calculations, losing incredibly easy marks. Just don't do it. Warning number 2. Failing to rigorously check specific terminology. And warning number 3, which we've really hammered home today, providing answers that are totally generic and not stamped to the specific context given. Listen, careless mistakes like these will absolutely destroy your grade boundaries, even if you perfectly understand the core concepts. To help you avoid freezing up and forgetting those complex enzyme roles during the exam, here is a super quick, actionable memory hack. Create a first-letter mnemonic. For example, for ligase forming phosphodiester bonds on Okazaki fragments, just think little penguins order fish. It sounds a bit silly, but turning complex processes into a simple, memorable sentence like that ensures you always have the right terminology queued up in your brain when that exam timer is ticking down. So, you've mastered the theory. You have your mnemonics. You know you have to apply context. But what if your biology is absolutely perfect and you still lose the marks? What is the hidden cause? Well, applying the perfect framework means absolutely nothing if you misread the exam's critical command words. If they ask you to evaluate something and you only describe it, your perfect biology knowledge simply won't save you. This creates an urgent problem that you absolutely must solve before exam day. You need to know exactly how command words dictate the mark band you can actually achieve. Understanding this is your mandatory next step. So make sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE, to master this in our next explainer. Until then, ask yourself this. Are you actually answering the question they asked, or are you just answering the one you memorized? See you next time.